So hi there, everybody. My name is Professor Lindsay Hall, and I am the Microbiome Group Leader at the Quadram Institute in Norwich, but I've also recently moved to the Technical University of Munich, where I'm Chair of Intestinal Microbiome. So what I want to do today and kind of during this session is to give you a very kind of broad overview and an introduction to the microbiome, with a specific focus on the gut and the microbial communities that live in there. So I'll just get started. So just first of all to say that I've got no conflicts of interest to disclose, but let's start off by delving right in. So the first thing to say is I'm actually a microbiologist by training, and I'm constantly amazed at these complex microbial communities that both live on and within the body, and how fundamentally important they are for our overall health and well-being. And just to give you some facts and figures behind these, I know some of you are probably quite familiar with the microbiome, but I think these figures are kind of mind-boggling and really important to share. So initially, we thought that our microbial kind of cells outnumbered our cells by a factor of 10 to 1, but more recent calculations have indicated that actually it's more like 1 to 1, but that's still a lot of microbial cells versus our own human cells. And I guess what's probably more important is it's not just about a numbers game here, and that actually probably equates to these microbes weighing a whopping 1 kilogram, which if you think how tiny microbes are, that's very, very impressive. But actually it's more important about the genetic material they encode. So what we're talking about here is the microbiome. And this, we obviously know about the human genome, but the microbiome is all of the genetic material that these microbial communities encode, and actually estimates for that reckon that it's about 150 times bigger than the human genome. So I think you can begin to appreciate how important these microbes are and all the additional functions that they encode within their own genomes, again, with respect to our overall health. And the other thing that's really important to say is that each of us harbors a very unique microbiome. It's unique to us. That does make studies relatively complicated when we're looking at humans, but obviously there's model systems that we can also use to kind of unpick the microbial complexity. But for the rest of the talk, I'm just going to be focusing on humans. And I think what's important to say here, and I mentioned this, these microbes live both kind of on and within the body, is that depending on where you are in the body, you get very different microbial communities. So for example, you obviously get, as probably everybody knows, we have these microbial communities in the nasopharynx. We obviously have an oral microbiome. And we know that the stomach, although it's with a low pH, means that not very many microbes can potentially colonize there, but we do have specific microbes that do, like Helicobacter pylori, for instance. The urinary tract also has a potentially normal microbiome as well, not just associated with urinary tract infections. The lung, which was for many years considered sterile, now appears to harbor I guess, low diversity but stable microbiome. In terms of women, the vagina also has a microbiome that's critically important for health, but also for pregnancy and pregnancy outcomes. But I guess in terms of all of the body sites studied so far, the gastrointestinal tract is really the kind of the most densely colonized ecosystem within and on the body. And it's also the best well studied. And that's really what I'm going to focus on for the rest of the presentation. So how do we actually find these microbes, and how do we look at the complexity associated with these different body sites, including the gut? Well, it really is kind of the advent of DNA and RNA sequencing that's kind of opened up the black box of complexity with respect to microbiome science. So, and this is done in a number of different ways. So what we can do here is we can do something that's called 16S Amplicon sequencing, which targets one of the key kind of, I guess, genetic components in prokaryotes. The ribosomal subunit, the 16S one. And these can be almost used as a molecular clock to understand kind of evolutionary diversity between different bacterial taxa. And in this case, what this will enable us to do is get down to kind of bacterial genus level. So it gives you the group of bacteria. It doesn't necessarily allow you to understand what the species is, for example. The next kind of one up with respect to kind of the more sequencing and computational based approaches would be shotgun metagenomics. And instead of targeting a very specific gene, so instead of just looking at the 16S ribosomal RNA subunit, what we do here is we actually look at the culmination of all of the DNA present in, for example, a stool sample. And what we can do is we can match these DNA reads back to specific genomes or microbial genes. And this enables us to kind of define species level resolution. And this would also, not just looking at bacteria here, we could also look at viruses, fungi, and archaea, for example. But importantly, what this approach allows us to do, metagenomics, is also get down to the function. So what's the functionality of these microbial communities? And then metatranscriptomics is kind of looking at, I guess, the RNA that's actually actively being kind of transcribed at any given time point. So moving up from just looking at the DNA sequence, what this actually tells us is what genes are actually switched on under a given situation. So this is kind of the next level up. And we aren't just kind of restricted to, 16S amplicon sequencing, shotgun metagenomics, and metatranscriptomics. There's other kind of functional omics readouts that encompass metabolomics, so looking at these metabolites these microbes are actually synthesizing, and also proteomics, which is looking at the protein level. So traditionally what we would have done with microbiology is culturing, but actually, and that kind of, I guess, fell out of vogue because we couldn't kind of culture, or we assumed we couldn't culture all of the complexity in the gut microbiota, but actually recent studies have indicated that that is not the case, and you just have to kind of roll up your sleeves and get on with it with respect to culturing. And if you use diverse and rich agar media, you can culture numerous different bacterial taxa. So culture omics is something that's actually very important in the field. And of course, having a culture is very important from a therapy perspective as well, because you need to physically have a microbe if you want to actually take that into the clinic. So as I've mentioned, I'm going to focus more on the gut microbiota. And this is, as I've mentioned, is because the gastrointestinal tract actually represents, well, it's not just the most densely colonized ecosystem in or within the body, but actually globally. So it's pretty impressive that we're carrying around these incredibly complex microbial communities just in our gastrointestinal tract. So I mentioned in terms of the complexity, well, at the moment, we expect that there's about 2,500 bacterial species that are capable of colonizing the colon, and that number is ever rising. And at the moment, that would kind of be more of the culturable bacterial species, but these kind of computational bioinformatics approaches that I mentioned previously have also indicated that actually it's probably tens of thousands of species. But if we think about the gut as an environmental niche, like I guess the rainforests or the oceans, depending on where we are in the gastrointestinal tract, the physical and the chemical kind of milieu is very, very different. So actually the microbes that prefer the upper small intestine, for example, so kind of lactobacillus is found in the small intestine, that's not the same microbes that we might find in the colon, for example, where bifidobacterium would be a dominant microbiota member. And if we just think about the bacterial taxa that are found in the gut microbiome, they kind of belong to four main groups, what we call phyla. So we have proteobacteria, we have actinobacteria, we have firmicutes, and we have bacteroidetes. And these are kind of so all of the, I guess, genus and species that I will be talking about belong to these four main phyla. And it isn't all about bacteria, although they are the best well studied. We also have the virome, and in fact, estimates reckon that that's about 10 times more complex than the bacterial side of things. Fungi are also incredibly important, so the microbiome, and also archaea as well seem to be an important component of the gut microbiome. So it's just important to say that it's not all about bacteria. There's other microbes in there that are also very important for our overall health. So we have these microbes, they're incredibly complex, but how do they develop over time? Well, actually, we've kind of got this ecosystem within us that is constantly changing and developing, right from when we're first born up until older age, when we kind of move into kind of more elderly years. So right across the life course spectrum. And what's important here is to say that there's multiple different factors that can actually change the composition of these microbes in the gut, which may have a positive or negative impact on our overall health. So let's look at this in a bit more detail. So this is just an overview, and if you remember the 4 main phyla that I mentioned on the previous slides, this is just kind of a pie chart showing how these microbial communities change as we age. So when we're born, we're considered to be sterile, and it's really only at the point of birth that we start to pick up our beneficial microbes. So we kind of get that first initial seeding with, I guess, what we would maybe call keystone or founder microbial species or strains. So infants have a very low complex gut microbiota where the ecosystem is just finding its feet and developing, which also means it's normally very vulnerable to external factors. So by the time we're about 2 to 3 years of age, we actually have more of an adult-like or quite stable gut microbiota. So kind of what you would see in toddlers. So the number of bacterial species has increased, as well as kind of the fungal component and the virome. And we see lots of kind of microbial interactions at this time point, which is also very important in the context of diet. And I'll talk about that later. As an adult, we have a relatively resilient gut microbiome that stays relatively stable, but if we do longitudinal analysis, we can see changes happening day to day. And again, external factors play a huge role here at shaping the microbial communities. And then actually kind of in older years, what we see is we kind of see a bell-shaped curve. So low diversity, higher diversity as adults, and then we start to see a decline in the number of bacterial kind of fungal components and virome in older people, and this is actually been linked to frailty indexes as well. So as just to say that, you know, these microbes are with us for our whole entire lives, but they're changing every day and as we age through different life phases. So just to kind of drill down a little bit more into the early life period, because that's what we do. We're particularly interested in my team and understanding how microbes develop during the early life developmental window. And actually, this is the first kind of 1,000 days is really perceived to be incredibly important for these first microbe and host interactions. I mean, we know obviously from a physiological perspective, this is when there's so many things happening kind of metabolically, kind of cognitively, immune development. This is all happening at these key life stages. So you can imagine that the microbes that are also colonizing and interacting with host cells during this time point is really important as well. So what's important to say here is that there's a number of different factors that might influence the gut microbiome. This is just a review that we did kind of just last year. And birth mode would be one of the ones that would be potentially linked to changes in the gut microbiome, which kind of makes sense because you get vertical transmission of maternal microbes to babies during natural childbirth, but this is interrupted during cesarean section. And in fact, the microbes that these infants will come into contact with might be skin-associated microbes or indeed hospital-associated microbes. Diet also plays a really important role with respect to the early life microbiome, which kind of makes sense because we're moving from a milk-based diet, and that might be breast milk or formula. And actually breast milk contains very specific complex sugar that preferentially feed certain bacterial microbiota members, including bifidobacterium. And formula does not have all these complex sugars called human milk oligosaccharides. So formula-fed babies normally have far less bifidobacterium, for example. And antibiotics, as I've mentioned, the early life microbiome is rapidly changing, the ecosystem still finding its feet. So there's a kind of overly negative impacts for external factors like antibiotics, which, as well as obviously treating serious bacterial infections, the issue here is that the antibiotics don't discriminate potentially between the good and the bad microbiota members or pathogens. And this means that we might even get some microbes kind of being killed off and then them disappearing completely from the developing early life ecosystem. So the next thing that we're just going to cover is what do these microbes actually do for us? So we're carrying these microbes around every day. We've got this extra kind of kilogram or even 2 kilogram in weight. So what do they do for us? And this goes back to my point about the microbiome and all of this genetic material that they encode. So the first thing that's really important to highlight, and kind of, I guess, something that maybe quite understandable in terms of there's an obvious connection here, is that the gut microbiota is incredibly important for infection resistance. And this is something that is termed colonization resistance. So this is when kind of your resident gut microbes actually make sure that any other pathogenic microbes coming into the gut cannot take hold and then cause infection. And this was actually described back in the 1950s. And this was some studies done in germ-free mice, so germ-free animal models. So these animals have completely free of microbes. And it was very clear here how important colonization resistance was, because there was an incredible lower amount of salmonella needed to cause serious infection and even mortality in these animals when there was no other resident gut microbes present. So what are the mechanisms underlying colonization resistance? So this happens in a number of different ways. And what I'm just kind of focused on here with a little schematic is kind of direct competition. So of course, the gut microbiota would be expected to be actually, competing with potential incoming pathogens for nutrients. And this would be one way to potentially exclude them from the gut environment. Another thing that is really important is actually these microbes are capable of producing novel antimicrobial factors, including bacteriocins, that represent very small selective peptides that kill very specific pathogenic microbes. And there's unsurprisingly a lot of research happening in this area with respect to developing new antibiotics with the rise of AMR. And the other thing that's really important is just physically, the gut microbiota will be covering particular areas in the gut. That might be the gut lumen, it might be on bits of food that are transiting down, it might be in the mucus layer that kind of overlies the gut epithelium, so that single cell layer in the gastrointestinal tract, that basically means that pathogenic microbes cannot get access to the underlying kind of mucosa and then potentially cause infection. So what's important to say here is colonization resistance is incredibly important. However, these mechanisms don't necessarily discriminate between pathogenic invading microbes. They also are an issue for more commensal microbes. So this is some of the same issues that taking a probiotic, for example, would also come across if you were taking them daily. And I'll talk about that just at the very end of the presentation. So the next one that's really important to highlight is the gut microbiota with respect to immune development. And it's really important to say here that that's not just important for the mucosal immune system, so the local immune system in the gastrointestinal tract. We're also thinking the priming systemically, so in body sites that are obviously kind of extra-intestinal. So other mucosal sites like the lung, for example, but these microbes are very important for programming the kind of splenic immune responses and, kind of basically inducing immune cells being produced in the bone marrow, for example. And of course, what we also want to think about here is boosting tolerance responses. So we want kind of normal, homeostatic or healthy mucosal kind of programming. We obviously don't want the immune system kind of, reacting to either self-antigens or food antigens, for example. And this is an issue obviously with different allergic responses. But we do also want immune systems to be primed, for example, anti-infection responses as well. So some of the work that these gut microbes do is they actually increase the mucus layer, which obviously acts as a physical barrier, which I've already kind of mentioned for microbes, but other pathogenic microbes coming in. They produce specific metabolites, including short-chain fatty acids, and I'll talk about that in a moment, because these come from fermentation of specific dietary substrates. So an example here would be acetate, butyrate, or propionate, and these are known to actually directly signal specific immune cells and kind of basically get them programmed. We also get a strengthening of the epithelial barrier because these microbes produce signals that means that the epithelial barrier is tight, because we don't want a leaky barrier because that's associated with numerous different kind of disease conditions. We get kind of enhancement of immune receptors so that when something potentially does come into the system, they're far more reactive. And then basically the microbiota programs every single immune cell that you can imagine. So T cells, dendritic cells, B cells and antibody responses, including secretory IgA, mast cells, natural killer cells, T regulatory cells, and the list goes on. So these microbes are absolutely fundamental for programming our immune system. And as you can imagine, this is why they're incredibly important, but why many people are looking at these in the context of therapy development. But I know that there's some other presentations that will cover this in a bit more detail subsequently, so I won't go into this in too much detail. So the next one is to look at the microbiota in diet. So this little kind of diagram here, feed me in terms of your gut microbes. Of course, whatever you eat also influences what microbes are able to then metabolize or ferment in the different regions of the gastrointestinal tract. And in fact, obviously we take in simple sugars into our diet, and most of these will be absorbed in the upper small intestine, but we also take in more complex kind of carbohydrates or non-digestible substrates that we can't actually break down ourselves, and actually it's our resident gut microbes that do that job for us. And as I've already mentioned on the previous slide, some of the, I guess, the end products of this fermentation process would be short chain fatty acids, and they're important for immune responses, but they're important for a whole host of other physiological developments and also kind of day-to-day processes as well. And they also produce kind of key amino acids and also vitamins as well. So we can also take a vitamin supplement, or we can just have a microbe that basically synthesizes that for us in the gut already. So you are what your gut microbes eat, so kind of whatever comes into the system. So normally we have these microbes that we consider primary degraders. So these are kind of the microbes that take over and start digesting these complex kind of polysaccharides, carbohydrates, dietary substrates initially. And then they basically break them down into smaller kind of dietary components, smaller sugars, that the next kind of microbial member might start to degrade. And these are kind of cross feeders. And this is very much can be seen as an ecological network. So you can imagine this is unbelievably complex in the gut, but is also incredibly important and interesting to study. So the microbiota might start, certain microbiota members like Rose Burrier, for example, might start digesting fiber, for example. And then based on this kind of fermentation, they will produce these metabolites, including short chain fatty acids like butyrate. And butyrate is actually incredibly important because it acts as an energy substrate for colonocytes in the colon, so strengthening the epithelial barrier. But we're not just restricted to we're not just restricted in the gastrointestinal tract. This kind of obviously has massive impacts for brain regulation, but again, I know we've got another presentation, so that will be covered in detail. Of course, kind of metabolism in the liver as well, and also of course for kind of fat deposition and adipose tissue as well. Again, it's incredibly complex. And I think the whole way through, although I've not mentioned it so far, apologies, there's a number of papers that I've highlighted that might be of interest if you want to kind of explore these different concepts further. So we have these microbes that are incredibly important for kind of day-to-day health. So this is one of the reasons that we obviously look at the microbiota, as unfortunately in many disease cases, alteration in these gut microbes have been linked to kind of either increased risk of a particular disease or disorder, or for actually kind of exacerbation of symptoms, for example. So some of the causes that might disrupt these resident gut microbial communities, and I mentioned a few of these with respect to early life, would be diet. So, kind of a high fat diet versus a low fat diet, not taking in enough fruit and vegetable and not getting these kind of, fiber and complex carbohydrates that beneficial microbes like to kind of ferment and digest. Antibiotics, I've already kind of mentioned them, not being able to discriminate between kind of good or potentially pathogenic microbes and how that might mean that we get almost extinction-like events. Birth mode, I've already kind of touched upon that. Infections, so of course, the microbes that come in and cause disease like Salmonella are also always constantly evolving, and they've come up with a number of different strategies that actually will change the microbial communities, which might actually augment their ability to cause infection. Genetics, there's a number of studies now coming out indicating that certain kind of traits in the human genome might also influence what gut microbes are there. And there's a number of other things like, kind of environmental factors, but I won't touch upon them. So, these are, and drugs would actually be another one just to mention that. So this is kind of just an overview in terms of how all these different external factors might influence the gut microbial communities. And these are some, and this is not an exhaustive list, of the diseases that have been, and I've put this, associated with microbiota disturbances. Okay, so we need to kind of think of cause or consequent, and this is why it's really important in the microbiome field that we're moving beyond kind of just descriptive studies, but actually understanding the mechanisms underlying them, because that's fundamentally important for therapy development. So, you know, intestinal diseases, which kind of make sense because you're local in the gastrointestinal tract. Metabolic diseases also kind of, I guess, quite a strong link there, but also importantly, autoimmune diseases or allergic type diseases, which links back to, you know, programming of the immune system. Brain link conditions, again, there'll be a fantastic presentation on that later. And again, kind of these allergic type diseases as well. So this is why it's really important to look at the gut microbiota, or in fact, other microbial communities, because they're linked to so many kind of diseases. And of course, most of these diseases have been going, you know, rising in the last kind of, 15, 20 years, and that's potentially linked to changes in the gut microbiota. So what's important to say, and I think this goes back to the colonization resistance point I said, is that unfortunately we kind of have these microbiome disturbances which are linked to, you know, these changes in the gut microbiome, and which may increase risk of disease or, you know, kind of initiation of disease pathogenesis. But altering the microbiome is, we have to think of this as an ecological problem. So, you know, this is kind of, you know, the gut lumen, we have the rainforest. So if we're kind of selectively wanting to kind of change diet or put in specific microbes, this is really quite a tricky proposition. This is not, you know, an easy thing to do. This is why we need to think of this kind of more holistically. So in terms of opportunities for health, and again, I'm not going to touch upon this too much because there's another presentation covering this in more detail, but these are obviously some of the, I guess, physiological kind of properties, conditions that we could think about for microbiome therapy, you know, anti-infection, thinking about metabolism, so just looking exclusively in the gut, the immune system and the gut-brain axis. And I think this is probably important to think about in the context of microbiota therapies, but also in the context of diet. So the ones that I kind of guess most people will have heard of would be kind of probiotics versus prebiotics. You know, probiotics are what's available kind of, you know, in the supermarket, either in a yogurt or maybe in a capsule. And these are obviously live microbes that actually go directly into the system. And the whole idea is that they potentially give a health benefit. One of the issues at the moment is that there's many probiotics in the market, but they don't actually have health claims. Or prebiotics, and these are actually specific dietary components that selectively feed specific beneficial microbiota members to actually then improve health outcomes as well. But if you think about it, probably a combination of both of these is really what's needed, because if you give a probiotic and it doesn't have something it likes to eat, it's not going to colonize, and it may not then give you a benefit. So it's again very important to be thinking of this ecologically and thinking about how we can manipulate the system. And just finally, just to touch upon, I guess, some of the more, I guess, recent kind of endeavors in the microbiome therapy area, which is again looking at specific dietary manipulations. There's a lot of work looking at the Mediterranean diet, for example, in different populations, including kind of in the elderly, and improving kind of healthy aging. Novel drugs, so actually the gut microbiota can actually augment or inhibit the activity of certain drugs that we take. Metformin is a good example of this. This is very important to think about stratification of patients for responders and non-responders. Diagnostics, for example, so thinking about infection, but also even in terms of thinking more at biomarkers. Is there a microbiome signature associated with somebody maybe having a higher risk of developing a disease in the next kind of couple of years? We obviously were fixated on humans here, but this is kind of more of a One Health approach. So, you know, we need to be thinking about how some of the microbiome therapies might also be important for kind of animal health, and some of the actually what we're learning maybe in the veterinary side of things might be as equally important with respect to humans. And developing targeted, so instead of probiotics, single strains or even maybe 2 strains, thinking about microbiota cocktails, so, you know, communities of microbes that potentially might benefit our health. And this basically stems from the work looking at fecal microbiota transplantation, which is incredibly efficacious for antibiotic refractory C. diff infection. But of course, if we could refine fecal microbiota transplantation to identify only those communities of microbes that are beneficial, this would mean that it would obviously be potentially kind of safer from a regulatory perspective, but hopefully we could also get kind of even better outcomes in these patients. And this would be something moving away from probiotics, but something called live biotherapeutic products. So, I mean, Our team is obviously doing a lot of work on this, but there's a huge amount of work going on this at the moment. And that is my final slide. I realise that's been a whirlwind, and hopefully I've not crossed over too much with some of the other kind of webinars in the workshop that you're going to hear. But hopefully that was interesting, and very kind of happy to chat to other people if anything kind of piques your interest and you want to chat further about understanding the microbiome in a clinical context. And just thank you very much for listening.